So as we saw before, this is just a summary of what we've already seen. Glucose goes through glycolysis to become pyruvate. The pyruvate is then pumped in across this inner membrane and allowed to diffuse through facilitated diffusion in between the two membranes. Once it gets pumped into the inner membrane, it then goes through its first oxidation step into acetyl coenzyme A, which then gets fed into the Krebs cycle. And remember, the whole process here, the goal for it is simply to produce ATP. We're burning ATP at such a high rate, we have to reproduce that ATP, and glycolysis does produce some. We get ATP that's made through that process that we saw before. The thing is, though, in order to pump this in, we basically use that ATP up. The Krebs cycle, if you remember, also produces a little bit of ATP, not very much. Remember, there's that one step where we make the GTP first, then the ATP. But this is not producing very much ATP, ultimately. This is gone, and this is a very, very small amount. So that's not really what these things are producing. Glycolysis and the Krebs cycle are not really all about that. What they're about is this. They produce NADH and FADH2. They reduce those NAD and FAD into these reduction products. Those are then fed into another process that occurs inside the mitochondrion that's called the electron transport chain, which is coupled to a third process that is called oxidative phosphorylation. So it's this. This is really where the majority of your ATP is coming from, electron transport at OXPHOS. What has to happen, though, is that these products then are going to be further oxidized. They're going to be oxidized again. The energy is then going to be used to make the ATP. To do that, what happens is the energy on NADH and FADH2 make this mitochondrion into a battery, a very real sense battery. That battery is discharged, and as it discharges, it makes the ATP. So let's take a look at that. That's what I want to see. Where I want to go first is right here, right along this inner membrane. If you see that inner membrane here, that's where the enzymes of the electron transport chain and oxidative phosphorylation are found, just on that inner membrane. So in this picture then, here's the outer membrane out here, the mitochondrion. Here's the inner membrane. And in that inner membrane are embedded a bunch of enzyme complexes. And these enzyme complexes form the electron transport chain, which is going to do essentially what its name says. It's going to transport electrons down this series of complexes. Now, these enzyme complexes are again floating around in this membrane. They're not connected the way I've shown them here, but this is just conceptually how they're going to operate. The first one that operates is this one here. It's called NADHQ reductase. And the reason it's called that is because it's going to oxidize NADH and it's going to reduce Q. Q is this complex right here. It's short for quinone. The Q complex then will operate next by donating electrons to this called cytochrome C reductase. It's called that because it reduces cytochrome C, which is this complex here. And cytochrome C then is going to be oxidized by this, which is therefore called cytochrome C oxidase. These together, NADHQ reductase, Q, cytochrome C reductase, cytochrome C and cytochrome C oxidase, all of them together form the electron transport chain. Now, there are about 1 million of these complexes in each mitochondrion. Like I said, the complex parts are floating in the membrane. They're not connected like this. But again, conceptually, that's how we're going to be thinking about them, because the electron transport chain is going to be exactly that. This is going to transport electrons down from one end to the other. So, in our mitochondrion, we have an aqueous solution. And if you remember, aqueous solutions always have hydrogen ions, and that's what these black H's are. These are ions now, not hydrogen atoms. These are just protons, plus, H+. And these are floating around because in any aqueous solution, remember, water is a weak acid, so it always dissociates, and it dissociates a little bit. So we have hydrogen ions on both sides of the membrane because there's water on both sides of the membrane. Now what's going to happen, like I said, is that this complex is going to turn the mitochondrion into a battery. What a battery is a separated series of charges through a capacitor and a resistor, that's what the membrane is, that then can be discharged so that there is a large voltage difference across this membrane, and the voltage can be discharged, and that it can be used to do things, in this case make ATP. So here's what we're going to do. We're going to pump hydrogen ions across this membrane, and we're going to move the hydrogen from the inner portion to this space between the inner and outer membranes. Okay, so we're going to pump them into this compartment. All right, now, What mechanism should we use? Well, we can't use diffusion because if you look here, this is just not really accurate. There's not just 5 of these on each side, but there's 5 on each side here to represent that these are equal, the concentrations are equal. So there will be no diffusion, there can't be diffusion. So there has to be active transport. We're going to actively transport these across this membrane. Active transport, as you know, requires metabolic energy. Now, in the active transporters that we've seen so far, the metabolic energy was delivered from ATP. In this case, we're not going to use ATP for a couple of reasons. First, the whole purpose of this is to regenerate ATP. There's no point in using the thing you're trying to build to drive it. And the other thing is we have these high energy compounds. NADH and FADH2 are very high energy compounds, more high energy than a single ATP. So what we're going to use is the NADH, and I'll show you what NADH does, and I'll show you what FADH2 does in just a second. But here's what happens. The NADH comes over here to the NADH-Q reductase, and it gets oxidized. What that means is, again, it donates electrons. The electrons then get transferred to the NADH-Q reductase. Okay, and so now this has been reduced. These are high energy electrons. They're coming off the NADH, and they're carried at a very high energy. Since they're high energy, they can go through a series of reactions that have a negative delta G that will transport them down the electron transport chain, like this. A series of redox reactions. I'm not going to go through all their details. You will when you get your upper division class, maybe. And you're going to see then that these are all just oxidation reduction reactions where these electrons get donated from place to place. They go from the NADH-Q reductase to Q. The Q donates it to cytochrome C reductase, which of course reduces cytochrome C, which is then oxidized by cytochrome C oxidase. By the time it gets down to the end of this chain, the electrons are at a very low energy state. Okay, so this then is releasing free energy. All of these reactions have a delta G that's negative. Since these are all exergonic reactions, we can use them to drive the endergonic reaction of moving the hydrogen ions against their concentration gradient. And that's exactly what we're going to see. So the electrons on this pump 2 hydrogen ions on average across here, and then they get donated to the Q. The Q then donates the electrons to the cytochrome C reductase. Cytochrome C reductase also appears to pump 2 or so hydrogens across the membrane. The electrons then get onto cytochrome C, and they're donated then from cytochrome C to cytochrome C oxidase. And the oxidase then appears to pump only 1. That's the current hypothesis. Now, there are a couple mistakes that you're going to see in textbooks and are still repeated. And this that I'm showing you is sort of the best we can do in understanding exactly what's going on. But it's still not exactly clear what's happening here. But it appears that this is only pumping 1 hydrogen ion across for every 2 electrons that go down on average. Now, there was a mistake that's perpetuated in textbooks still today that say all of these each pump 2, so 2, 4, 6. It appears really to be 2, 4, 5. Okay, now that becomes important when we actually do the final accounting of everything. But that's what's going on. That's what the electron transport chain is doing. It's taking these electrons and literally transporting them down this chain using the energy that they release to pump hydrogen ions across the membrane and make this battery. Okay, so again, the whole purpose of this is to make ATP. Before we see how that works, though, we need to take a look at a problem. We have to solve a problem, and that is this. We started with 2 electrons in the electron transport chain at a high energy state. They lost energy as they transported the hydrogen ions across. Now here they are sitting on the cytochrome C oxidase at a low energy state. We have to get rid of them. We've got to get them off of that. Because if we don't reoxidize the cytochrome C oxidase, then what's going to happen is all of these electrons are going to back up and the whole process comes to a halt. The battery gets discharged, and once the battery is discharged, the mitochondria become useless and they cannot regenerate the ATP. So we need some way to get the electrons off of here at a low energy state. The problem with that is that most things will not accept electrons at a low energy state. The only things that do accept electrons at low energy state are things with a very high electronegativity. So we have to go to the periodic table and look for something with a high electronegativity. And if you remember, in the upper right-hand side of the periodic table is where the high electronegativities tend to be. There are 2 different elements that we could use that are not too reactive, not too reactive with electrons, but are reactive enough that will take these electrons off. And those two are sulfur and oxygen. Now, some living creatures use sulfur to do this. We use oxygen. And ladies and gentlemen, here's the connection between the Krebs cycle and oxygen use. The Krebs cycle is producing the CO2, but here's where the oxygen is used. The oxygen is used to accept these low energy electrons that came off of the NADH that ultimately came from the sugar that you ate. And the oxygen then picks up both of those two electrons. One oxygen picks up two electrons and adds two protons to make water. Interestingly enough, this process also detoxifies the oxygen. Oxygen is a deadly, deadly poison, which is why these antioxidants are a good thing to have. So it's taking the oxygen in a deadly form and making it into a benign form of water. So this is how the electron transport chain works. High energy electrons come off of NADH, comes down through the electron transport chain, end up at a low energy, and end up being put onto oxygen. And that's where you balance the equation when you oxidize the sugar. Okay, so all of that then is the electron transport chain, and all it's done is made the mitochondrion into a battery. So now the question is, what do we do with that battery? Well, remember, the whole point is to make ATP. So how do you do that? Well, let's go back to this. Here we have our electron transport chain. Now, it's not the only thing that's embedded in this inner membrane. There's a bunch of stuff. And another thing that's in there is this. This is a channel, an ion channel. But it's an ion channel specifically for protons. And it is named ATP synthase. In fact, ATP synthase is part of this channel. Guess what ATP synthase does? Of course, it synthesizes ATP. So the point is that this is the battery we're going to make. We're going to discharge the battery through ATP synthase, and that is going to be how we construct our ATP. All right, so let's go through all of this. Our first step is this. What we first do is we run the electron transport chain by taking an NADH and pumping 5 hydrogen ions across the membrane. There we go. So we had 5. Now let's keep the accounting. The 5 hydrogen ions then get pumped across, and what happens then is I have this concentration gradient, which I'm going to allow to be relieved through here. What happens then is a hydrogen ion then will go through here, and a second one will go through here. So 2 hydrogen ions will diffuse through this ATP synthase, and when it does so, it drives a mechanism that catalyzes the formation of ATP. And again, remember, going from to make ATP, we have to start with a DP and a free phosphate. So it takes a phosphate and an ADP, makes it into ATP, and this discharges enough energy to do that, which means it's discharging enough energy that it's at least 7.3 kilocalories per mole. So 2 diffuse through, and this process then is going to make the ATP. So ADP plus a free phosphate. The phosphate is a phosphate that is in your diet, and it gets into the mitochondrion and is used then to make the ATP. So this becomes the gamma phosphate on this ATP. Okay, so that's the counting. Approximately 2 hydrogen ions will make a single ATP. Okay, so all of that's beautiful. Now, we still have a concentration gradient though. Remember, we pumped 5 across, and now we've only let 2 go through, so there's still 3 extra over here. All right, so now I've got an ATP. I can let 2 more hydrogen ions flow through. So 2 more actually diffuse through here, and I do the same thing, take an ADP and a free phosphate, and I make ATP out of it. Okay, now notice I still have enough gradient here to allow another hydrogen ion to pass. But once that 1 hydrogen ion's gone through, now everything is equal. Okay, so let's go back up here. You can see I pumped 5 across, 2 went through, made an ATP, 2 more went through, made an ATP. That's why there are 4 here. So there's 1 extra on this side. There's a total of 6 on the outside here. When 1 flows through, that gives me 5 on the inside, 5 on the outside, and everything is equalized. So that means that I cannot get enough energy here to make another full ATP. Okay, now, the way I've shown it to you is highly, highly simplified. And that simplification is something that I need to sort of get past at the moment. Here's a simplification. I'm showing you only one of these complexes and one ATP synthase. In the mitochondrion, remember, there's about 1 million. Every single mitochondrion has about 1 million of these complexes. All of them are operating at the same time. And it's not just one NADH that drops off its high energy electrons, it's a bunch of them because there's a bunch of glucose that's constantly going through glycolysis, constantly driving the Krebs cycle. So what happens then is this, think of it this way. Another NADH comes across here, pumps 5 more hydrogen ions across. So now I got 5 plus the one that was already here, that's 6. 6 hydrogen ions will allow me to make 3 more ATP. Okay, because if I have 6 here, 2, 4, 6, and I get 3 ATP, and therefore I get 3 plus the 2 that I made originally, that gives me a total of 5 ATP. So that means this, I get 5 ATP for every 2 NADH, right? 2 NADH gives me 5 ATP, or if you wish, and we often will say it this way, 1 NADH will give us 2.5 ATP. So that extra hydrogen here represents 1/2 of an ATP. And that's where you're generating the ATP. Now remember, you're making a bunch of NADH. In fact, what I would like you to do before you take the quiz is count up how many NADHs you make, how many FADH2s you make, and how many ATPs you make net through 1 mole of glucose. Starting with one mole of glucose going through this entire process. How much NADH, how much FADH2, and how much ATP will you ultimately end up making. Now, I want you to count up the total number after you take all that NADH and figure out how many ATP you're going to make. And you know each NADH is going to give you on the order of 2.5 ATP, or every 2 is going to give you a total of 5. What about FADH2? Well, FADH2 operates like this. Here is a picture that comes out of your book. Here's the electron transport chain. The yellow arrows show the electrons going down the electron transport chain, getting put onto oxygen at the end. And the red arrows show you the transit of hydrogen across the membrane. Here they're being pumped, and here they're diffusing down through the ATP synthase to make ATP. So this is oxidative phosphorylation, and this part is the electron transport chain. Okay, and then we got 2, 4, 5, and that's why we get the 2.5 ATP for 1 NADH. Now, FADH2 is carrying its electrons at a lower energy state. FADH2 feeds its electrons in here. It skips the NADHQ reductase. So what happens is then the electrons then go here, then here, then here, and then they go on oxygen. So here, instead of 5 hydrogens being pumped across, only 3, 2 here plus 1 here. And so from 1 FADH2, you get 1.5, or if you wish, 2 FADH2s will give you 3 ATP. Okay, so now you can do the accounting on your own. If you count up all the ATP that can be made from all the NADH and FADH2 and straight ATP that are made from the Krebs cycle, glycolysis, and pyruvate oxidation, you should be able to come to the proper number. Now, I'm going to ask that on the quiz, so that's going to be one of your quiz questions. One of the interesting things you're going to see is that there are a number of mistakes in textbooks, and they get the number too high, so don't look it up. If you look it up, you're liable to get the wrong answer. And that's because people made mistakes when they originally wrote these textbooks and thought that this gave the same amount of these others, or I've noticed a lot of textbooks ignore the ATP that's used to pump the pyruvate across and so forth. So you need to be careful to make sure that you follow through what we have here in the lecture. Now, the interesting thing is you're going to get a number that's pretty high. The number that you get is higher than any number we've ever measured in real cells. There's a number of reasons for that. I'm not going to get into all of that, but a lot of what I'm saying is if this system works perfectly, then it'll do exactly what I said, and it doesn't always work perfectly. Also, there are other ways for the system to get discharged without making ATP. For example, when you have a high hydrogen concentration, you can have some leak out of the mitochondria and into the cytosol. So you lose some of the potency there as well. So the best we tended to measure is around 20 to 25 ATP, which you're going to find is a number that's lower than the number you get. If you get a number that's lower than 25, you got to recount. So there's a lot going on here, and it's not as efficient as one might hope, but it is efficient enough to keep you alive. And it is absolutely critical. You're dependent on it. You yourself, not all organisms are, but you are completely dependent on this. Here's how we know. Cyanide is a very rapidly acting poison. All cyanide does is it blocks this last step here in the electron transport chain. It blocks the use of oxygen and will not allow these low energy electrons onto the oxygen to make the water. It just blocks that. And if it does that, you don't die next week, you don't die tomorrow, you die now. And the reason is for what we just said. Each cell is 2 billion ATP, 100 million per second you're burning. You got 20 seconds left of ATP. If you block this system, you're going to die in less than a minute, or at least the cell will die in less than a minute. So that's absolutely critical. That's why it's absolutely critical. Now, the last thing here is I just want to take a look at a close-up, sort of a cartoon close-up of this ATP synthase. If you look at the ATP synthase, this is essentially a reasonably good cartoon of what's happening. There's still a lot here that we don't quite understand, but here's what we know. When the hydrogen ions flow through here, it causes this portion of the membrane to rotate relative to this portion down here. This is the catalytic portion, the part that sticks off here. It's called the knob. And it somehow is able to use that motion of the rotor in the membrane to catalyze the reaction and add energy, the energy required to lift this substrate level phosphate and put it on ADP to make ATP. So there's still a lot going on here, and we still don't know all the details. You want a Nobel Prize? Figure that out. Figure out exactly what's happening here, because that's one of the key questions that we still have in molecular biology, one of the many, many questions that we still have in molecular biology.